Biology, Chapter 19, Theory Video 11. So now we realize that there's a lot of information stored in these genes, and we can find out a lot of details using these mechanisms that have been developed. So a single DNA chip would give up about 10,000 pieces of information about the presence or absence of any genes in the genome of what we are going to check. So all of these details are collected in a lot of databases, and this we there should be a possibility for us to collect this data, process, analyze these biological data, so that we can we can advance in the other things that we find. Now, for example, if we do a if you find a new genome sequence, or if you find or go into more advanced step, or if we find the genomes of another species, now we have to compare these things. So computer databases and computer softwares have been developed, and these computer softwares are known as bioinformatics. So this is the collection, processing, analysis of biological information and data using computer software. So bioinformatics is quite useful in dealing with these things. I would suggest that you read through this, because this is not something that should be explained. This is something that you need to read and understand, but I will explain whatever it is that needs to be explained. So what happens is, there are different kinds of databases established throughout the world, but it is all maintained in the internet. So all the researchers can access these things. However, there are authorities who have the ability to add and change details that are in this. So we can actually hold this information, and we can compare using the details that we have. So now, for example, Ensemble is a database that holds the genomes of eukaryotic organisms. There would be many others. Now, for example, genomes of zebrafish, mice. So these are used to compare the genomes of human beings, and because they are used widely in researches. So they are used, their details are used, and they are publicly available. So we don't have to actually start from the scratch, but we can develop from whatever has been already found. Now, there is a search tool called BLAST, that is Basic Local Alignment Search Tool. So that has an algorithm which compares the primary biological sequence information, and it will allow us to find similarities between sequences that they are studying, and with the ones that have been already saved in databases. When a genome has been sequenced, or when their sequences have been identified, scientists are able to compare those details with the values that have, the genomes that have already been saved in these databases. So they can actually find out their similarities, and thereby they can understand what is what genes are responsible for which proteins, etc. All of these details can be found by scientists using these details. So this is a home page of RCSB protein data bank. So there are many different websites and databases that provides information, or collects this data and provides information for us to compare. So people are able to have access. At least the research community can use them. However, there are some databases where not only the research community, but other people also can use. CFTR2 database, it holds all of the 412 variants so far identified of the CFTR gene. So it is, it will not only provide, the information needed for healthcare professionals, and it will actually help the to people to understand the mutations that can cause cystic fibrosis. And also it the publicly making these databases available would allow people to do research um, on developing new drugs and vaccines for different diseases, and they have been proven helpful in many different ways. Write down a note on this. Digital biology handles big data. Bioinformatics is the process where the collection, processing, and analysis of biological information and data using computer software is used. When genomes have been sequenced, comparisons can be made with the use of sequences that are already been saved in these databases. The database Ensembl, ENSEMBL, holds data on the genome of eukaryotic organisms. Search tool BLAST, BLAST capital, is an algorithm for comparing primary biological sequence information. And the details that have been identified by microarrays and comparing primary structures of proteins and visualizing the 3D structures of proteins are all a possibility because of these databases. This allows for researchers to read gene sequences that would provide them information in developing vaccines and understanding biological phenomena. Some databases provide information to the public at large, going beyond research community, and makes and allows people to understand new advancements that have been made in healthcare industry, and having up-to-date information on discoveries such as mutations that can cause cystic fibrosis. I hope you understood. If you have any questions, please ask. Please read this through once again. There is nothing for you to understand, just for you to memorize some of these things. Just have a general idea, that would be sufficient. You don't have to know the names of these things or the search tools. You should be able to explain why they are valuable using the note that I gave you.